So this is ASTO5 project, finding and characterizing variable stars in the Andromeda galaxy, difference image analysis. So these are the interns. The one in the middle, that's me. My name is Nana Pukwa. I'm in grade 12, going to grade 13. I'm a Ghanaian and I'm 17 years old.
Hello everyone, I am Mariam Al Khouri, 16 years old from Egypt.
Hi everyone, I'm Praisha Keval Ramani. I'm 17 years old and I'm from India.
Hello everyone, I'm Pembo Song, or you can call me Billy. I'm grade 11 student from China.
Hi everyone, I'm Jingting Shen. I'm grade 12 student from China.
Hi everyone, I'm Srujanika Kapil Rao, a grade 12 student from India.
Hello everyone, I am Rudra Pratap Nandi from Bangladesh and grade 12 student.
My name is Benedict Baden. I'm a student in grade 11 in Ghana.
Hi everyone, I'm Jocelyn Cortez and I'm the primary mentor for this project. And my secondary mentor is Ali Patel. He's a PhD student under our faculty member, Professor Raja Paragra Bohatvakarta.
So the introduction, okay. ASCO5, our mentor who had been taking us was Jasmine Fortis. We just saw her. So this project focuses on uncovering the secrets of variable stars, stars that change in brightness over time, within the Andromeda galaxy, our closest galactic neighbor. The reason why we study Andromeda is because it's a perfect place to study stars since we can't observe our own galaxy from outside. Studying the Andromeda galaxy gives us an external perspective, like looking at your sibling in the world without mirrors. The Hubble Space Telescope, the largest digital image taken by it ever of the galaxy, provides a sharp, high resolution view across a vast area with stellar brightness captured in six filters across the UV, visible light, and infrared proportions of the electromagnetic spectrum. Variable stars are the key understanding of their internal processes of stars, revealing insights into their structure, evolution, and life cycle. When a star brightness changes, it acts like a cosmic heartbeat, providing astronomers with a rare glimpse into what is happening deep inside. Some of these stars even serve as a cosmic distance markers, ha- helping map the scale of the universe. Next slide. The following tasks that we did in this project was labeling variable stars, carefully examine different images to mark the positions and important details of stars that changes in brightness, documenting documenting these brightness changes, record how the brightness of the stars change over time, plotting on color magnitude diagrams, um, help to plot the label stars on the color magnitude diagrams, which help us to tell more about their properties, understanding their PSF, and learn about the point spread function and see how its changes can impact the appearance of non-variable stars in the difference image. Next slide. So, what is difference image? Difference image in astronomy is created by subtracting one image from another at a different time, obsulating the changes like variable stars, transits, or moving objects by removing constant background features. Um, so how does it? As I said, we take two images from the same part or the same area, but in different time intervals. So then we align them together in the same position and we subtract one of the image from the other. And that's where we get the difference image because this is the difference between both of the images. Um, what you see is that the unchanged stars or the objects disappear because you remove any background features. And the brightened objects are the one that see as bright spots. The dim ones are the one that we see as dark spots. And the transit of sources are the one that we see very clearly. Next slide.
And now we are talking about importance of difference imaging. Difference imaging is an essential technique for detecting variability in densely populated stellar environments, such as star-clustered and M31. In such crowded fields, traditional photometric analysis is often limited by blending and source confusion. DI overcomes this by subtracting images that taken at different epochs, isolating genuine changes in stellar brightness. DI allows accurate confirmation of variability, even when light curves are sparse or noisy, by detecting residual flux at the star's position. It plays a key role in distinguishing real variable stars from artifacts caused by cosmic rays, bad pixel, or noise in the data. The identified variable stars can then be linked to the physical properties of their host clusters, such as age and metallicity, allowing estimation of each star's initial mass and evolutionary phase. The eye also helps us assess the confidence level in the measurements by considering the impact of photometric blending in crowded environments. Next slide, please. We have talked a lot about variable stars, but what is this definition? Variable stars are stars that exhibit changes in brightness over time. These variations can result from intrinsic factors such as pulsations, eruptions, or evolutionary changes in stars' internal structure, or extrinsic factors like eclipses by a companion star. In stellar clusters, variable stars serve as powerful tools for studying stellar evolution and pulsation mechanisms, since their physical parameters can be constrained using the well-known properties like age and metallicity of their host clusters. This research leverages data from PHAT survey to identify and characterize variable stars in M31 clusters, enhancing our understanding of their evolutionary stages and initial masses.
So here's some insights on the last project we did. Begin with in the Andromeda Galaxy M31 section, there's a catalog of 86 bright variable stars in star clusters using archival Hubble Space Telescope data from the FAT survey. It analyzed sparse light curves and used difference imaging to identify the stars, then match them with theoretical models to determine their evolutionary stages and initial masses, ranging from 0.8 to 67 molar masses, to 67 solar masses, sorry. They also assessed how blending in crowded cluster environments affects measurement confidence. The stars span a wide range of types and phases, including Cepheids, red supergiants, and Wolf-Rayet stars. 12 stars were characterized with high confidence. This is the first systematic study of variable stars in galactic clusters using Hubble, therefore highlighting the value of clusters in studying stellar evolution and variability. Next slide, please. So, the final legacy version of the panchromatic Hubble Andromeda Treasury, which is a FAT survey photometry, has been released, featuring reprocessed Hubble Space Telescope data across ultraviolet, optical, and infrared bands. The new method combines all overlapping exposures and uses CTE corrected images, improving star detection and photometric accuracy, especially in NUV bands. Although more resource intensive, it significantly reduces systematic errors and improves data completeness. The updated catalog now includes 138 million stars. That's an 18% increase from the last one, hence enabling high quality stellar population maps that reveal mixed older populations and structured younger ones.
Labeling software and video. Rudra and Praisha. So Astro Data Lab and GitHub. Introduction. Astro Data Lab provides a powerful platform to access large astronomical datasets like Phantometric Astronomical Survey, and our mentors set up a GitHub repository to make it easy for us to work with them. By simply downloading the FATS packets through the provided scripts, we can start labeling stars without worrying about complex data preparation, code and files. The GitHub repository includes FAT packets through the provided scripts. We can start. Please, next slide. The GitHub repository includes ready-to-use Python scripts, configuration files, and example notebooks for downloading and labeling star data. These files handle all the setup automatically, so we can focus on analysis rather than manual file handling. Next slide, please.
By running the provided notebook cells, we can download the FAT packets directly into our working directory within minutes. This process ensures we have the latest, correctly formatted dataset ready for labeling and further study. We use specialized labeling tools to mark stars in each image, ensuring accurate and consistent annotations. All labeled images are stored in the detections folder, which organizes our work for easy access during analysis and reporting. We will now show you a video as to how we actually label the stars. First, we begin by opening the DI labeling file and launching the DI labeling tool. After running all the code, we select the specific cell we want to label stars in. My computer then automatically downloads the relevant FAT packets. Once that's done, I run the kernel, which loads the image I'll be working on. The tool displays 3 versions of the image: the template, science, and the difference image. For labeling purposes, I use difference image as it highlights changes between the two. I zoom in and identify a star I want to label. To mark a star as real, I press R, which highlights it with a green circle. If I believe the star is bogus, I press B, and it gets marked with a red circle. There's an additional tool called D, which means delete when I want to remove a label. I open the logs and the detections folder. This is where all the stars are automatically stored.
This part, we are going to talk about our future work. We'll have 3 parts in this section. First, the color magnitude diagram, the most direct application of the different imaging tool. And the second part is intersection with machine learning. And the final part is the relationship between the different imaging tool and other programs in the SIP program. So first, the color magnitude diagram is obtained from the photometric data we get from the DI labeling tool. And in order to make a color magnitude diagram, we should first detect and label the various stars based on their brightness variations across epochs and ensure that there are enough data from at least 2 photometric bands. And the graph in the left is an example of the color magnitude diagram from a research paper in 2019. And the first step to make a color magnitude diagram is matching. So after we obtain the photometric data, we can match the coordinates of variable stars identified by the DI labeling tool with those in the photometric catalog. And this ensures that each variable star is associated with accurate magnitude measurements in at least 2 photometric bands. Accurate coordinate matching is crucial to avoid misidentification of the variable stars. Next slide, please. And the next steps are calculation and plotting. So first, once the photometric entries are confirmed, we can calculate the color index for each star by subtracting the magnitudes of two different bands. And in the plotting part, we can plot so the magnitude in the redder band is used for the vertical axis when we plot the color magnitude diagram. Slide, please.
Second parts of the future approaches will discuss how human-generated real bogus DA labels can help to train a machine learning model. So in the first way, in general, it can help machine learning in three ways. First, as training data, let the model learn to identify variable stars. Machine learning models requires a large number of label samples to learn the patterns. The positions of variable stars we marked will become the samples for the model. By learning these labels, the model can gradually master the features of variable stars in star maps and ultimately achieve automatic identification and marking of variable stars. Next slide, please. The second way is for model evaluation and optimization. After a model is initially trained, it needs to be tested with label data that has no incorrect answers to evaluate its performance. Our manually labeled results will serve as the standard answer to be compared with the model's output to calculate metrics such as accuracy and omission rate. By analyzing where the model went wrong, we can in turn optimize the model's algorithm or parameters to improve its performance. And the third way is build high-quality datasets to promote research in the field. Machine learning in the field of astronomy is highly dependent on high-quality label datasets. If our manual labeling results are organized and standardized, they could become a small but professional datasets. Such datasets can not only support the iteration of certain models, but may also provide basic data for other research teams in the future. So all in all, the manual tagging work we're doing now is paving the way for the automation of subsequent machine learning.
Lastly, we're going to look at how this DI labeling project about variable stars connects to two other SIP 2025 research projects, ASTO1 and ASTO5. To recap, ASTO5 focuses on finding variable stars in the Andromeda galaxy using difference imaging to check variability in stars' brightness through the DI labeling process. This data is enhanced by machine learning, where humans provide the truth samples to account for cases of bad subtraction or instrumental effect, helping develop machine learning with manual inputs so that variable stars can be found throughout the survey and not just in areas we've inspected. ASTO1 can use this data set to train other methods or models, excluding the DI labeling tool to identify variable stars. ASTO4 compares the relativity between weak CN stars and Cepheids. For context, between three galaxies, M31, M33, and LMC, during a certain phase of stellar evolution, stars exhibit certain unexpected and unusual chemistry in their atmospheres. A presence of a chemical molecule, CN, which is carbon and nitrogen, is found on the surface of the stars. This phenomenon occurs in massive stars when they have evolved past the main sequence and have become red supergiants. These stars are classified as weak CN stars since the effect of the CN has a very subtle signal, and the rarity of red supergiants created a gap in the data available regarding this occurrence. Now, carbon and nitrogen are known to be produced in massive stars, though the exact reason and process is still unclear. However, this happens in the core of stars and not on the surface. Research is trying to understand why CN is found on the surface of these stars. Experts suggest that as the stars pulse, movement of the internal contents causes the CN produced at the core to travel to the surface of the star. However, this is only speculation and no concrete evidence of this has been found. The presence of CN in both Cepheids and in weak CN stars led to the question, are these stars in fact the same? ASTO4 established that Cepheids are a distinct class, separate from weak CN stars, since they fall in a different region of the color magnitude diagrams. However, it is reasonable to assume that the connection may lie in the evolution of these stars, suggesting that the massive stars go through the Cepheid phase first, where they pulse, pushing CN to the surface, and later evolve into red supergiants or weak CN stars. ASTO4 and ASTO5 analyze the same sets of stars and data, only using different methods complementary to each other. One major way in which ASTO5 contributes to research in ASTO4 is through sanity checks. Using the DI labeling software to check if the Cepheids that have been identified by other groups using ground-based data are also seen as variable by us in the software. Verifying that the stars are in fact variable gives people extra confidence in the data collected due to the use of two separate surveys and telescopes. Finding stars in the data set that are not variable still adds value, showing the percentage of previous detections that are legitimate and those which are spurious. One thing to keep in mind, Cepheids have a 4 to 10-day variation period. So if images compared in the difference imaging are taken closer together than this range, we may not be able to provide any significant information about the variability of the star. The second and more direct way in which ASTO5 contributes to ASTO4 is through the check of variability of weak CN stars, since red supergiants are not known to be variable. To sum it up, ASTO4 focuses on the relation between weak CN stars and Cepheids by looking at spectra and color magnitude diagrams. Since ASTO4 uses datasets from other groups, ASTO5 contributes to the research by verifying the authenticity of these Cepheid detections. Finally, I would like to give a heartfelt thank you to my amazing teammates for their constant support and collaboration through this journey. Grateful to the SIP program for this wonderful opportunity to learn and grow. It's been an incredible experience working with such passionate and talented individuals. Thank you all.
I'd like to thank my fellow interns for their incredible support, collaboration, and the great memories we've made together. It was truly great to meet you all.
So I'd like to thank our mentors, especially from me, and then on the behalf of the team as well. Our mentors Jasmine, Alvi, and then Raja. Thank you so much for the time you've put in and the dedication for us. We hope to work with you again. Thank you.